quite diverse, and we used Campylobacter as an example of this. So bacteria evolve, and they can do so by generating variation to the genome at different rates. So for example, some species are slower at evolving than others. So an example of the slowly evolving species is Yersinia pestis, and on the other hand, a faster evolving species is vancomycin-resistant Enterococcus faecium. So bacteria can accumulate genetic variation into the genome by two different mechanisms. So they can either take up DNA from their environment and incorporate this into the genome through recombination, through horizontal gene transfer, or they can accumulate point mutation through DNA replication errors. And this accumulation of molecular change over time can also be termed as the molecular clock. And an example of in the lab where we've seen in real time bacteria evolving is the image to the right, where E. coli were evolved to become more resistant to higher concentrations of antibiotic over time. So why is it important to understand rates of nucleotide change in bacteria? So it's important for understanding evolutionary events and adding timescales to these events.
So for example, understanding pathogen emergence and spread, virulence, AMR, vaccine escape, and also for tracing outbreaks or dating host jumps. And two examples of studies where rates of nucleotide change have been incorporated into the study are in Streptococcus here and also in Yersinia pestis. However, good rate estimates depend on a good sample set, and estimates can be improved by, so if there's a large time difference between comparative strains over which to calibrate a mutation rate in the first place, lots of isolates belonging to the same lineage, and also enough sampling of lineages that represent a whole species. However, though, most estimates are based off the availability of samples at a particular time, and also a lot of estimates aren't representative of entire species due to limitations in sampling. However, this is a lot easier in medically important isolates, where there'll be a lot of the same lineage sampled for clinical reasons. Also, more medically important infections have limited hosts, so there's a smaller host range to sample from. And also, time-calibrated phylogenies can be incorporated into epidemiological events. And for example, this study on the right used a time-calibrated phylogeny to investigate transmission in C. diff. But it's more challenging to obtain robust rate estimates in species which are diverse and environmental. And a good example of this is Campylobacter. So Campylobacter has multiple hosts, multiple reservoirs, multiple lineages, and also it does that. Some kernel complexes can have multiple hosts, so generalists, and they can jump from host to host, so it makes it more difficult to trace back infections. So because of this, it's difficult to obtain sample sets that are representative of the whole species in the wild. And also the information that we already have on mutation rates have mainly been based off MLST. So we have tried to address these challenges using sequencing of archived isolates. So we had a collaborator who had some Campylobacter isolates in their freezer from the 1970s. So we sequenced these and that gave us an extended sample time frame. And also we looked into whole genome data sets, so we used the Campylobacter BIGS-DB and obtained lots of different genomes from lots of different hosts, sources, and clonal complexes. And we decided to use a microevolutionary approach to estimating mutation rates of nucleotide change. So after we collected the large genomic data sets, we assembled these and built trees. And then we looked within these trees to identify closely related pairs of isolates. And they had to have a long enough time difference between them, but they also had to be close enough to share recent common ancestry. And it was a very simple approach to estimating mutation rates from these pairs. So we quantified synonymous, non-synonymous nucleotide changes, and then this included from within recombination and from without recombination events. And then we used the time difference between the two between the two branches belonging to the isolate pair, along with the nucleotide change difference, and worked out different rates of nucleotide change based on this. And we did this for all of the pairs selected from the tree, and averaged the particular rate of nucleotide change over all of the pairs for C. coli and jejuni separately. So we use synonymous mutations excluding recombination as a molecular clock, and we could compare this to previous estimates only for C. jejuni, but this was based on MLST. We found that our rate was significantly lower than previous estimates, but the important rate of nucleotide change for further analysis was the total rate of nucleotide change, which included all variant effects from including and excluding recombination events. So we then wanted to see how we could use the total average estimated rates, both for C. coli and C. jejuni, and use these to infer common ancestry through time. And we did this by using the birthday problem as inspiration. So the birthday problem is a mathematical problem where you think about how many people, random people at a party, and within all of these people attending the party, how many of them can share the same birthday? So what's the chances that two people at this party would share the same birthday? And we wanted to apply this to ancestry so out of all the strains at different time points in our sample sets, what are the chances that at each point in time will isolates share common ancestry and eventually give rise to successful progeny in subsequent generations? So we developed the idea of the effective lineage size, and an effective lineage is based off traditional effective population size, NE, but it's more relatable to clonal organisms like bacteria. And what it basically means that at each point in time, there's a population size, and there'll be certain lineages circulating at that time. And an effective lineage will be the lineage that will have successfully occupied a niche and will then go on to divide and give progeny to the next generation. And if you look at the diagram here, the effective lineage are the black dots, and the grey ones will go extinct. So this will be the unsuccessful population size. So what can the effective lineage size tell us about bacteria? So it gives an idea of the number of successful ancestors at a given time point, which will give rise to lineages in successive generations. It will give an idea of niche availability or occupancy at over time, and also the maintenance of lineages over time. So if a bacterial species has been successful, they will have a higher number of maintained lineages over time. And also we can use it to measure the extent to which a population has diversified and sort of put this onto a time scale. So using coalescent theory and estimated total mutation rates from the first half of the analysis, we developed an equation of how we can work out the effective lineage size. And I'm not the best at maths, but we use the equation Z equals Y divided by X. And Z are the effective lineages, which is the same as ancestors, so successful ancestors at a given time point. And this is also, if we're tying it back to the birthday problem, it's the equivalent to a shared birthday. So Y is the every potential pair possible from that data set. So for example, for C. coli, there were 601 isolates in the tree, and to get every potential pair, we did 601 squared divided by 2. And then we divided this by every possible pair at a time interval. And then we did this equation for every time point across the sample time frame for each species. And we plotted this number, so the effective lineage size number for both C. jejuni and C. coli. And there's still relatively short timescales of evolution for bacteria, but it was the longest sample time frame that we could achieve. And we can use this to compare differences in diversification or lineage diversity over time for both C. jejuni and C. coli. And we found there to be a much lower maintained number of lineages over time in C. coli than C. jejuni. And this can be interpreted as C. jejuni occupied more niches in the past than C. coli did. But also, on the other hand, the diversification for C. coli was much more rapid in recent years than C. jejuni. So in the past 8 years into the sample time frame, there was an increase of ineffective lineages of C. coli by 800%, which was almost 3 times more than C. jejuni. And this could be linked to colonization of agricultural niches, and C. coli diversifying much later on than C. jejuni. So in conclusion, our approach highlights the importance of sampling when calibrating rates of nucleotide change. It provides an alternative way of obtaining more robust rates in recombinogenic or environmental species. It's a novel approach of exploring diversity in populations, and this can be applied to other opportunistic pathogens, which is something that we're working on. I'd like to thank everyone for listening. I'd like to thank Ben Pascoe for inviting me to speak in his session. Thank the Shepherd Lab that I have spent the past 4.5 years working with, and also all of the collaborators who we've worked on this project. And I'd be very happy to take any questions. Thank you.
Thank you for that talk, Jessica. It was really interesting. I really enjoyed listening. We'll just go to a few questions from Discord. We have one here from Ben. Which other organisms do you think this method would be good for?
So we are working on a project on this. And we wanted to compare different organisms with different transmission ecology. So, for example, multi-host, more environmental organisms like Campylobacter compared with more obligate pathogens. And because they have different host ranges and host numbers, do we see differences in the effective lineage sizes over time?
Okay, thank you. And another question. The estimates for the molecular clock are more robust using your method, which based on genome pairs that have diverged very recently, I'm not mistaken, but how do you apply them to slightly more distant genomes as then most signal comes from multiple mosaic recombinations? The divergent state would then be overestimated.
Yeah, so our molecular clock estimate is based off synonymous mutations outside of recombination events. So because in Campylobacter, recombination is the major source of variation. So we removed these and just looked at mutation rates with the remainder of the genome. And a lot of noise is introduced into Campylobacter genomes when estimating mutation rates in traditional methods. So we wanted to use the closest related pairs as possible to remove this noise.
Okay, and maybe just one more question from Desi. I might have missed this, but what sources are these samples coming from and how diverse are they for C. coli and C. jejuni?
So we wanted a diverse selection of genomes as we could possibly get. They come from pretty much every host we could find in the database, which is a lot. So it's environmental and poultry and a lot of pinnacle cases, and also the same with the kernel complexes. So we took a good variety of representative kernel complexes, so the specialists as well as the generalists as well.
And thank you, Jessica. There's maybe one or two more questions in Discord if you would like to get to those later, but we are a bit pushed for time, so I will move on to our next speaker.